Welcome to this lecture on asexual reproduction in bacteria and other prokaryotic organisms. In this particular lecture, I shall be focusing on the minor types of reproduction, namely different exospore formation, including budding, including cyst formation, and then I shall be talking about the asexual reproduction in cyanobacteria, that is the different types of endospore formation, and then I shall be going into Archaea and we will see the different types of asexual reproductive mode in Archaea. The first important role of bacterial reproduction is the exospore formation. You can see in this particular slide that is the exospore is in the form of thick-walled spindle-shaped spore, which is normally found in rhodomicrobium, and this particular spore is produced in chain and they are actually held by filamentous structure with swollen spindle-shaped body. This swollen spindle-shaped body is formed of thick peptidoglycan layer. It is without any dipicolinic acid, and at any point of time they get separate and they can produce a new bacterial cell. And this is the exospore in photomicrobium. But definitely during exospore formation, the photosynthetic activity is suspended because the metabolic rate is too low. So this particular structure is helping them to survive during unfavourable condition. This phenomenon of budding is found in planktomyces, where you can see there is a central cell and there will be stalk-like projection, and this stalk-like projection will be producing peripheral swollen structure which is commonly in the form of buds, and these buds actually will reproduce, the DNA will reproduce and they will produce minor structure or small offsprings, and these offsprings will be separating from this particular stalk-like projection and will produce new cell. But this particular structure is the prostheca, and this prostheca is the stalk-like projection of the vegetative cell. So very often this particular prostheca will indicate whether that particular bacteria will produce an endospore or whether it will develop exospore in the form of buds. Now the particular life cycle of Colobacter is actually showing these different stages of binary fission and the stalk formation. As you can see in this particular slide, that the different stages of cell division represented by the gap 1, the synthetic and the gap 2 phase is actually showing what are the changes that take place in the bacterial cell of Colobacter. What we see is this particular cell with the flagella and the pili is the original bacterial cell, and the DNA will gradually show methylation or hemimethylation and it will prepare itself for division. And then during the initiation of the S phase, this particular bacterial cell will lose the particular flagella and the DNA start replicating. So here you can see as it is going to the middle of the S phase, they are producing the long holdfast like stalk, and this particular holdfast will hold the bacteria to the substratum, and at the same time the particular DNA is almost getting fully replicated and it will continue in the next phase. And then during the outset of the G2 phase, there will be the formation of the FTZ ring, which will divide this particular bacterial cell into two cells, and the upper one or the daughter cell will produce the non-motile bud, and this non-motile bud towards the end of the G2 phase again will produce the active flagella, and then this particular bacterial cell will divide fully. One will be showing sedentary habit, that it will be attached to the substratum, while the other with the developing flagella will now be free living or swarming cell will be produced from a stalk-like cell. What is important in Colobacter are the presence of this stalk and the presence of the cilia. So there must be different protein factors which actually control this stalk and this cilia formation. You can see in this particular slide that this is a Colobacter cell and there are different factors represented by pli-C and pod-J. In the first case, here actually the pli-C is a histidine kinase protein, which is determined, whose polar position is determined by the pod J protein. So you can see it is actually this plici which ultimately is developing the cilia. But at the same time, the pod J, it is having the two forms. One is the pod JL. When the pod JL is there, there is no cilia formation. The moment the pod JL is truncated to produce the pod JS or the short form, then the cilia is formed. And at that particular point, the two cells separate. One becomes the swarming cell, one becomes the stalked cell. The same thing we see is in another case where the CCKA is the histidine kinase, whose position is being determined by the CTRA protein, and ultimately the same thing happens. Ultimately the grey cell with the cilia is given out as the swarming cell, and the white cell is sedentary with the stalk. In the same way, the div K and the div J, which actually determine the position of the poles. The div K will here determine the formation of the cilia, and the div J will determine the formation of the stalk. So there are definite protein factors which ultimately determine whether that particular bacterial cell will produce the cilia or whether it will produce the prostheca and thereby getting attached to the substratum. But what is interesting that in this particular case of Colobacter, that the stalk is actually the projection of the same cell and it is not a foreign structure. But in this particular stage, that is where we see in Gallionella, this stalk is not actually formed of protein. It is formed of some iron which is deposited, and it will produce a long iron stalk. And this iron stalk is definitely helping the bacteria to attach themselves in the particular substratum, which can be a pipe, which can be the floor of a particular pond. But this Gallionella in this way is doing the deposition of iron in their body, and thereby they are doing some sort of biomining.
Sir, I have a question. Iron being a heavy metal, does it impart any toxicity to the bacteria?
You see, whenever a heavy metal is getting inside the cell, it will impart toxicity. But the bacteria is clever enough. The bacteria is depositing into the surface protein. So it is projected outside the cell. So in no way it is imparting the toxicity inside the cell, or it is inhibiting the metabolic activity inside the cell. So thereby this particular stalk is giving support to the bacteria, and on the other hand it is causing damage to us, because sometimes this iron deposition may lead to clogging of pipes, because this iron will be deposited on the surface and the bacterial colony will block the water flow in the pipe. Now cyst is another type of bacterial reproduction which is commonly found in Azotobacter, or here it is in Rhodospirillum. Now this particular Rhodospirillum cell, you can see gradually it is showing elongation and it is showing deposition of stored food. It is in the form of poly-hydroxy butyrate. This white shaped structure is the poly-hydroxy butyrate, and they will then divide inside a thick exine. And now you can see first there are 2 cells, then there are 4 cells inside a common thick outer cyst wall, and this cyst wall is also nothing special. This cyst wall is formed of peptidoglycan. Of course, when we see in case of eukaryote, there may be other components like in chitin or in form of other protein which form the cell wall. But bacterial cell wall primarily is neither formed of chitin nor it is collagen, but essentially it is a structure formed of peptidoglycan. But what is important here, that these cysts will deposit the nutrients, will remain there intact during unfavorable condition, and with the advent of favorable situation they will germinate and they will produce the new cells, and these cells can be swarming cells or the cells which are swimming away from the existing cyst. And what is important, the cyst will remain as empty shell, and this is called the husk. So in this particular Rhodospirillum centenum, you can see this unique life cycle which is alternating between cyst and the swarm cells. Primarily, it is survival during unfavorable condition. At the same time, it is depositing the stored food in the form of poly-hydroxy butyrate, and there may be sometimes it so happens that this cyst will help the bacteria to survive for longer period, and thereby it can actually increase the pathogenicity. When it is inside my body, this thick wall will protect it from gastric H cell or other digestive enzymes. This metabacterium is a filamentous or elongated cell, and at the time of asexual reproduction, it is actually increasing in size. And as it increases, you can see that they develop segmentation, specific segments. And as these segments are formed, they are producing a typical segmented filamentous bacteria, and this SFB will produce the small cells inside. You can see these are the cells which are produced inside. And ultimately these cells will undergo swelling and will produce functional endospore-like structure. So this particular structure actually starts in the form of a filamentous growth and ultimately produces endospore, and thereby it allows the bacteria to survive both during unfavourable as well as during favourable conditions. The cyanobacterial cell is actually dividing simply by binary fission like in normal bacteria, and this process, the cells are called the macrocyte. But always the cyanobacteria will not reproduce by binary fission. Instead, they will grow in size and they will produce internal cells. And these internal cells or endospores are commonly known as baeocytes, and they are commonly found in Pleurocapsa or Staniaria. This particular cell will produce definite thick-walled green cells which look like the vegetative cell, only they are smaller in size. And they ultimately will be enlarged, the DNA replication will be there, and definitely they will produce an aggregate of cells in the form of a colony, and ultimately the parent cell will break, the baeocytes will come out and they will germinate to produce the normal cell. But there is a definite question that what is the primary difference between a normal baeocyte and a macrocyte. As I have already told you, the macrocyte will be bigger in size, the baeocyte will be smaller in size, and definitely the macrocyte, the normal metabolic activity will be fast regained. But in baeocytes, they are more like resting spore, so they will take more time to be normal again. So definitely macrocytes are produced whenever the vegetative condition is ideal or there is adequate food, and at the same time they will also photosynthesize faster because of the presence of the PS1 and PS2, whereas the baeocytes will take more time to show photosynthesis. Cylindrospermum you can see here, they are mainly filamentous cyanobacteria. Now Akinetes are basically thick-walled spore. They may be terminal or they may be central in position. And these thick-walled spore are produced more in number when there is more nitrogen. In the presence of, say, a compound called dinitrophenol, they are actually inhibited. So sometimes the condition is such that there will be more Akinetes produced, and sometimes when there is any antibiotic like chloramphenicol, like streptomycin or like dinitrophenol, this particular filamentous structure will not produce any Akinetes. Now Akinetes can be macrocyclic or microcyclic. Now microcyclic Akinetes are smaller one, and naturally their endurance power is also less and they germinate faster. Whereas the macrocyclic ones, they are germinating less faster and they can act as a storage cell for a longer period.
So what is the difference between an akinete and an endospore?
Yeah, you see, an endospore is commonly found in a bacterial cell, and this endospore is a thick-walled structure, multi-cell layer is there which will allow the bacteria to survive during unfavourable condition. But an akinete is a relatively thin, not that much thick walled spore where there will be normal photosynthetic activity, and most importantly this particular akinete will be flanking another type of spore which is heterocyst. So the photosynthetic activity in akinete is relatively more than in endospore. The akinete formation is also seen in Anabina cylindrica. This Anabina cylindrica were subjected to stringent environmental condition, and it is grown in a limestone rock because of its alkaline pH for 28 days, and it was subjected to very low to very high temperature. Maybe they are subjected to -80 to +80 degrees centigrade. They were exposed to UV. They may be 325 to 400 nanometer. They were given very low pressure condition, and under this condition they were allowed to stabilize, and then they were sent to the low Earth orbit with the help of a satellite, and they remained there. And this is almost 1/3 of the normal geostationary satellite, which is at a height of 22,282 miles. And when they were sent almost a month, they survived. But of course, they showed massive photo bleaching. This is the normal green condition where there was no photo bleaching, and this is the photo bleached Cyanobacteria. And when they were allowed to be again remain in the dark, they again developed the photosynthetic pigment. So that means the entire Cyanobacteria is not dead. The Anabina again is coming alive because of the presence of this thick-walled Akinetes, which allowed the photosynthetic activity, produce the chlorophyll, and again make the bacteria survive. So in one word, this Akinete is a reproductive cell because it helps in the rejuvenation of the cyanobacterial cell. Hormogonia is commonly found in a filamentous cyanobacteria. Like nostoc and anabina, they will show specific region which is like a disc-like structure. But actually it is not a disc. It is a portion where there will be no photosynthetic cell. And whenever there is some external pressure, then this particular fragment will be breaking, and they will break by fragmentation and will produce a new cell. And so this new fragment which is produced is called hormogonia. But this particular figure you can see in nostoc, this hormogonia can actually exhibit gliding motility on one hand, and also it can exhibit adhesion. As you can see here, these blue structures, these are stained hormogonia present in nostoc. So what does it indicate? It indicates that it can actually make the bacteria move from one unfavorable situation to another favorable situation on one hand, and at the same time when it reaches the favorable condition, it will stay there by adhering to the substratum surface. Hormocyst is actually a thick-walled structure which is developing close from the heterocyst, which is another thick-walled cell. But these hormocysts will actually produce new filamentous structure whenever they are breaking by fragmentation. And this hormocyst will produce new plant body. And the hormocyst definitely will allow the bacteria to colonize in a favorable condition. Fragmentation is the process, and it is the process by which the hormogonia or the hormocysts are getting separated from a filamentous bacteria and will produce new plant body. You can see in this case, these are the new filaments which have been actually fragmented from the parent filament and produce new plant body, and thereby in one way they are producing a colony of filamentous cyanobacterial cell. There may be another type of spore which is commonly called the exospore. You can see they are clearly produced from the tip of the filament, and there may be slight protection in the form of this gelatinous wall, and these spores are produced in chain. In case of oscillatoria, they are cut off due to a water current, and they produce new filament on being separated. And these exospores, because of their minute size, they are also termed as nanocysts. So these nanocysts or exospores in case of cyanobacteria are also a unique method by which they can colonize and they can survive in unfavorable condition. Heterocysts as such is a thick-walled spore which actually is helping the cyanobacteria to fix nitrogen because of the presence of nitrogenase enzyme. But this heterocyst definitely is being flanked by a unique cell which is Akinete. So Akinete is the photosynthetic component, heterocyst is the nitrogen fixing component. You can see initially this filament have the same type of cell. So there will be some of the cells, this is, you can see here it is showing gradual enlargement. And as the cell is getting enlarged, it is developing new material, and this new material will produce the mature heterocyst, and as they produce the mature heterocyst, it is not showing any photosynthetic activity, it is only involved in nitrogen fixation, and for that the nutrition is being supplied by the vegetative cells in between, which may be the Akinetes or other vegetative cell. The ultrastructure of heterocyst, you can see that the heterocyst is having the outer polysaccharide layer, then it is followed by the glycolipid, then the outer membrane, then the cytoplasmic membrane. So it is a four-layered protective structure, and then there is the cytosol which is having the nitrogenase enzyme for nitrogen fixation, and most importantly they are having a polar granule. This polar granule is having the cyanophysian granules. So these cyanophysian granules are the stored food. During this process of nitrogen fixation, the nourishment is provided by the cyanophysian granules, which will produce these white channels. So it will produce a sort of honeycomb-like structure, which is nothing but the gradual dissolving of the cyanophysian granules. Ultimately, this is the matured heterocyst. It's considered as a particular reproductive spore because it is also helping the particular cyanobacteria to survive during unfavorable condition. And here, when it is stained with fluorescent dye, this absence of red fluorescence will indicate that the heterocyst is matured enough because there is no pigment there. Photosynthesis is not taking place there, because whenever there is photosynthesis, there will be oxygen liberated, and this oxygen is not allowed inside the heterocyst.
Sir, is Krebs cycle involved in heterocyst formation?
That's a good question. Actually, you see, heterocyst, I told you the metabolic activity is less, and at the same time oxygen liberation is also less, so oxygen utilization might be there. So definitely that means oxygen utilization means Krebs cycle will be there. Now this Krebs cycle will produce a series of organic acid. One of them is oxaloacetic acid, and this oxaloacetic acid will produce the oxoglutarate, and this oxoglutarate deposition in the heterocyst wall before it is formed in the pre-heterocyst condition will allow the deposition of calcium. And at the same time, this oxoglutarate will also help in the expression of certain protein like the PAT-S or the heterocyst K protein. And the development of this protein or expression of this protein will allow the maturation of the heterocyst, and thereby we can say that heterocyst formation is somewhat related to Krebs cycle organic acid. Archaebacteria is one of the oldest member of living organism in this world. Archaea also reproduces by normal binary fission. That means the cell is getting elongated and there will be the formation of two daughter nucleoid or daughter DNA by the normal DNA replication, followed by this kink or constriction, which will be ultimately determined by the formation of the FTSZ ring. And this FTSZ ring will complete the process and it will produce the new daughter cell. And because they are producing two identical cells, it is called the binary fission. Apart from binary fission, some of the Archaea can also reproduce by multiple fission. In this particular case, a multiple fission is a cell where it will undergo repeated division and it will produce series of these archaeal cells. And they are producing a huge colony which ultimately appear like lobes of brain, human brain. And these particular cells, with the advent of favorable condition, will ultimately germinate and produce new cell. But the question is, this particular cell, as because they are producing a thick external wall and they are showing repeated division inside, that is multiple fission, they also help the organism to survive during unfavorable condition. Archaebacteria is not showing a filamentous structure always. Normally this round cell will show elongation, will show multiple lobation, and ultimately from this lobe they will undergo binary fission and they will divide, and thereby they will show this typical phenomenon of fragmentation. That means this small fragment which is getting separated from the parent cell will produce a new cell. So this is how fragmentation may also take place in Archaea. Another very interesting Archaebacteria is this Methanocoelius nigri, where you can see this is the vegetative cell like you see in yeast, this type of budding. So here also you can see they will produce a bud which is a protuberance, a lateral protuberance, and followed by the division of the nucleoid material and entry into the new cell. So this type of budding is also a common phenomenon commonly we encounter in case of Archaebacteria. And this particular bud formation will ultimately produce a new structure or new phenomenon which is called genotypic switching. Now, what is genotypic switching? Can you answer?
So, phenotypic switching must provide some form of sexual dimorphism in Methanocoelius nigri?
It is not sexual dimorphism, but it is providing structural dimorphism. In the colony, sometimes the phenotype, it looks like a single vegetative cell. It will show elongation, so it becomes filamentous. So some of them are filamentous and they produce the budding. So the phenomenon of budding will make the cell change its phenotype. Some of them will be filamentous, some of them will be round or oval in shape. Clear? Yes, the phenomenon of cell division in Archaebacteria is controlled by a unique group of gene which is commonly called the CDV operon. You can see in this particular slide that there are 3 specific CDV genes, the CDVA, CDVB and CDVC, and they are showing overlapping, and they are actually being purified and separated by SDS page. The proteins are separated and they are also detected. This particular protein actually will control the cell division, and they will actually impart the cell cycle control or cell cycle checkpoints. These cell cycle checkpoints will indicate that if the CDV operon could find that the cell is dividing normally, they will not stop the process. But if there is some damage, DNA damage due to UV radiation, the CDV will cut off the process and the cell division will come stand still. So thereby, though Archaebacteria is a very primitive group of organism, it can also show a specific mechanism by which the cell cycle can be regulated. What we have learned today are the different modes of asexual reproduction in bacteria, in Cyanobacteria, in Archaebacteria. We have seen how the exospores and cysts are being formed. We have also seen how the endospores and the other types of asexual spores is formed in Cyanobacteria. And finally, we have seen that how Archaebacteria reproduces by binary and multiple fission or by fragmentation. And most importantly, we have also seen that how Archaebacteria is reproducing, or how the reproduction by asexual mean is controlled in Archaebacteria through CDV operon. Thank you.